Gentlemen, I'm happy to be here today to share some information that we have on the use of the digital sequence information for analyzing the gene bank collection and making conservation related decision. The digital sequence information refers to the genetic information that is derived from the nucleic acid sequence such as DNA, RNA, and stored and analyzed using the digital technology. It can be used in a different way. And I just listed here some few examples that can be used to identify species, subspecies. We listened this morning in the presentation that there are lack of taxonomists in the world. We have less expert in that domain. So digital sequence information can play a role here, understanding genetic diversity, identifying duplicate, guiding crop collection management, and also breeding. With the integration of the digital sequence information in the gene bank management, we have opened a new opportunity for conservation, for research, and for sustainable use of the plant genetic resources, because it has enabled the gene bank manager to work more efficiently to identify genetic resources with greater precision and develop also by breeder to develop new crop varieties that are more resilient to changing environmental conditions. So there are several techniques for using DSI in gene bank management. You have the whole genome sequence, you have the targeting sequence, you have also different platforms that have been developed in the world that use those information also. And you have several future use of the digital sequence information on the gene bank management in terms of predicting breeding, improving the efficiency and the sustainability of the breeding program, identifying novel genes on allele, or conservation of a breeding program. So some application of the digital sequence information for analyzing the collection could be like species identification, taxonomy classification, phylogenetics analysis, population genetics, genome analysis. You can use genetic diversity, the analysis of genetic diversity to identify underrepresented species in the collection and prioritize the collecting effort for population that have less diversity. You can use it also to identify novel genes, for trait discovery in the seed bank, like I was saying, to monitor the changes in the genetic diversity of the population over time, and also on the phylogenetics analysis. So I just want to give some few examples where we were able to use this digital sequence information to identify some human error or some misclassified species. This error could happen in the gene bank collection. If you look at this graph, where we have several groups, we found that one wild species named Oriza Batai were able to plug with some cultivated rice. So we were able to identify those misclassified species using these informations. So from there, we were able to use to identify or to develop diagnostic marker that could be able to differentiate the species of in the African rice collections. So we have several types of markers we developed, and those markers are able to separate the African species compared to the Asian species, etc. So we can also use this DSI to identify all the species that you have in the collection. This is the case of rice, where you have Glaberrima, African rice, Asian rice, and the wild species like Batai and Longistaminata. You have several clusters, and you can also be able to classify them. We use them also for the sampling methodology for selfing crop like rice, to know if we, for the large scale characterization, if we can use 1 seed or 15 seed of a bulk of 5 to 10 seed. And based on the result, we were able to make a decision on how we should handle this large scale characterization of the germplasm. We also identify, use digital sequence information to determine the population structure and understand the diversity in the collection. So we have here the structure that we were able to determine, and in the specific subgroup also, we were able to see how the specific subgroup is structured. In these are African sativa compared to Asian rice, the rice domesticated in Asia, and to see how the African sativa are structured and compared to the Asian rice. So we also use this information to develop mini core collection. You know that the goal of the gene bank is to conserve the germplasm for sustainable use, mostly in pre-breeding and breeding, to develop new crop which are adapted to the environmental condition. So it's often very complicated to breeder to screen those large size of collection in their breeding program. Therefore, it's very useful to create what we call mini core collections. The size of the collection can be handled easily in the multi-location testing or for sterile phenotyping by the expert. So we develop several mini core collections for African species and also for African sativa, and they are very useful. They are now screened worldwide to identify new trait, donor for new trait. And so in the breeding program, those mini core have been used for this trait. I just listed few traits which are important. They occur in the farmer field to identify new donor, to identify gene, and to be able to develop improved varieties. So there are, I just wanted to also to share with you some centralized database where this information are stored. I took the example of National Center for Biotechnology Information, NCBI. They have all this information where scientists can go and use the information and apply them in their program. We also have Grameen database developed by Dr. Suzanne Makouche from Cornell University, very useful for the gray species and for scientists working on the genomic research also. So this information, digital sequence information, are also very important to curate the passport data. Like I told you, when you have some problem of taxonomy, you don't exactly know the species, what you have collected in the field. If you have marker information, then those marker information will help you to classify the genetic resources that you have collected and to minimize error in your passport data. So I just want to share also some publication that we made based on this information. And with that, I will thank you for your attention. Thank you.